thanks so much, Michael, for inviting me, and thanks to Carol Collier and Megan Mackey for organizing this as well. So I'm Stefanie Kroll, and I work at the Academy of Natural Sciences, and I'm just going to talk to you today about our three-tiered approach to monitoring for the Delaware River Watershed Initiative. And I just want to acknowledge my colleagues, Richard Horowitz, Roland Wall, Carol Collier, and John Jackson from Stroud. So I'll just start by giving those who may not be familiar with it an outline of the Delaware River Watershed Initiative, and then go through our monitoring, some preliminary results, and then how we're organizing this information in a database so we can communicate it back out to people. This graphic was built by an artist who heard about our initiative. Oh, can you hear me?
Yes.
Okay. All right.
I'm sorry, I just muted all the other lines, but I unmuted you, so you're live.
Okay, thanks. This graphic sort of illustrates our vision for the Delaware River watershed in 50 years and the partners that we're working with to develop this program. So the William Penn Foundation started the Delaware River Watershed Initiative. They brought together the Academy of Natural Sciences and the Open Space Institute for planning it. Then the National Fish and Wildlife Foundation also came in, as well as the Institute for Conservation Leadership. So those are the people here on these arrows. We chose geographies in which to work, which we call sub-watershed clusters, or clusters for short. And those are the Brandywine Christina, Schuylkill Highlands. This red is the Middle Schuylkill, Upper Lehigh, Poconos Kittatinny, New Jersey Highlands, upstream suburban Philadelphia, and the Kirkwood Cohansey Aquifer, which you can see is on the border of the basin. So the initial investment by the William Penn Foundation was to stimulate on-the-ground actions that were focused geographically with an aim of improving and restoring and protecting water quality.
Excuse me, just one moment.
Okay.
Steph, I want to make sure we're seeing the right image, the slide now.
It's still this picture showing.
Okay. Great.
Yep.
Thanks.
Yeah. So the investment goes to on-the-ground practitioners who are doing agricultural BMPs, stormwater BMPs, and land preservation, and the goals have to be to improve water quality. Here, the cycle of continuous improvement is that it's really important to the foundation and to all of us that we collect data. So we think through how to do this, we plan actions and monitoring, implement them, collect data, and provide information for adaptation of this program and similar actions. And if you want to hear more about the initiative, we can talk about more details in a little bit. Here, going to just go over the goals of the Delaware River Watershed Initiative and how the Academy supports them. So there are other partners also supporting these goals, but I wanted to give you an idea of how our actions are supporting specific goals. So targeted investment, we have been working on site selection and review, applying consistent scientific metrics, and sorry, this is a lot of text, so I'm not going to spend too much time on it. So here we do outreach to other groups and quality assurance. It's really important to us, to the foundation, and to everyone involved that this be a collaborative effort, which we've been doing through steering committees as well as other grant proposals with collaborators. We want to be able to test strategies of on-the-ground action and hypotheses and be able to assess the impacts. So for this, we're also doing these actions of data collection and data analysis. All of this so that the program can adapt as it moves forward and also over a longer time horizon. and so that eventually we can demonstrate scalability and transferability. One important part to this last point is that we received a Growing Greener grant to look at past restoration projects and how they may have affected streams or not, so that that can inform what's currently going on through the Delaware River Watershed Initiative. So it's supporting the investment on the on-the-ground actions. We're very actively engaged with the observational infrastructure. So we want to measure impacts of actions on water quality across scales. We want to understand these watershed characteristics in our sub-watershed clusters, and then also understand stream characteristics relative to the projects that are being planned. We're measuring and sampling multiple indicators using quantitative methods, and these sites in the map are all the sites that were sampled in 2013 and 2014, and we know that we alone could not get to all these sites, and so it's important to coordinate with partners and get as much geographic coverage as possible. One thing that was really important to us, Margaret Palmer and other restoration ecologists have published statements saying, we know what the scientific theory is and why we're doing restoration, but there have not been a lot of significant investments in measuring that so we can show the changes that we predict we're making. And so that's a really important part of the investment that the Foundation has made in monitoring to complement the on-the-ground actions. So we started in the beginning, before projects were planned to go in, just to take a look at our subwatershed clusters and what the in-stream characteristics of them are. These are just pages from a document we produced called the Cluster Characterization, which is on our website, where we took macroinvertebrate IBI scores from the Pennsylvania DEP, the Philadelphia Water Department, and NAQUA, and just used the quality ratings for each of those systems to show the stream quality. In the middle school, they do have some green triangles for good, but they fade into the green spaces, so it's not quite as bleak as it looks in this picture. So here, this gave us an idea, first of all the hard work that the agencies are putting in, but also where we're starting from the last time these streams were sampled. One question we've had from people is that there's a lot of monitoring going on by a lot of different groups, so what makes ours different and why do we need such a large-scale approach? And part of that is because our objectives are different from other people who are monitoring. So agencies that have to meet laws about streams reaching their designated use have to decide if they're impaired or unimpaired. We want to see some fine-scale changes in the ecosystem, so small shifts in the abundance and the density of insects, fish, and algae. We want to see those changes. We expect them to be small initially, so we want to be able to measure small changes and get a baseline that will allow us to make comparisons when there are small changes. So we're using methods and indicators to detect them in the short and the long term. And part of this too is how are the ecosystems changing over time, and what's the lag from actions to effects on the ecosystem. We're also working to build a database for our research questions and to make our data available to others. And in doing that, we need to make sure that we have consistent data sets with known data quality that people can make use of. We have a lot of monitoring partners. So here I'm just going to go through the different watershed clusters and who we're working with there. Again, these are just our monitoring partners. There's a whole long list of people who are doing the on-the-ground actions, working with landowners and making those deals on the ground. And so these are the folks that I'm going to show you now that just are helping us with the monitoring. And then we'll talk about get into the three-tiered system. So partners in the Brandywine Christina are Stroud Water Research Center, so they've been working for decades in the Brandywine Christina, and we work closely with them in project design, and they collect the macroinvertebrates, and their team of interns identify them, and I'll talk about that more. So this is the Brandywine Christina. What we're calling the Schuylkill Highlands is this light orange area, basically the French and the Pickering Creek and some others. So here we have Green Valleys, Berks County, Hay Creek, and Stroud working with us. In the middle, Schuylkill, which is this light bright green, we have Miller Environmental with volunteers, Stroud, and the Berks County Conservation District. On the upper Lehigh is this darker green. We're working with the Wildlands Conservancy and Moravian University. In the Poconos Kittatinny, it's the Nature Conservancy, North Poconos Care, and the Broadhead Watershed Association. In the New Jersey Highlands, here in blue, TNC, the Musconetcong Watershed Association, and the Wallkill River Management Group. In Philadelphia, we have a lot of watershed associations, which is great because we have a lot of people in a small area. So Tookonee-Tacony, Wissahickon Valley, Lower Merion, Perkiomen, and Pequesing, as well as academic partners at Villanova and Temple universities. And then in the Kirkwood-Cohansey, we have people at ANJEC working with Friends of the Black Run Preserve, as well as New Jersey Audubon and the Pinelands Preservation Alliance. So a lot of people, a lot of really enthusiastic people, and as we've been working with them, we've seen that the enthusiasm is across the board and the training levels are different. So the watershed associations may have people with training in stream ecology leading them, and then they're taking out a group of people that can be anyone who lives in the area. But then we have people like Stroud or Villanova or Temple who are pros at what they do and take all that scientific rigor into the field. And so that's why we decided we really needed to have a tiered system, because it's important to us that data are collected. It's important that these groups that are often involved with the on-the-ground actions can also incorporate their stakeholders and their communities in the work being done, especially to get the message out and to keep this going once the original funding runs out and we need to find more partners. So we are collecting multiple indicator sets. So this diatoms and soft algae, macroinvertebrates, and fish. And in this diagram, I just wanted to show some results from a study they did in Germany. This is for the response of these different groups to land use, and it's in different stream types. So we know that algae can respond to specific concentrations of pollutants, including organic pollutants, and they also can respond to flashy streams that scour them off. Macroinvertebrates are sensitive to overall organic pollution, habitat degradation, and broad-scale watershed disturbance. And fish are also sensitive to local habitat degradation and watershed disturbance. So by collecting all of this information, we can tell a more complete story and get closer to saying which stressors really are affecting this stream the most, so that we can start to tackle the non-point sources of stress. We have some different types of sites that we've sampled. The first type is called integrative, and these were chosen by John Jackson, Rich Horowitz, and Dave Keller before the program really got going. So the point of the integrative sites, and that's what's in the map here, is to capture the typical conditions within the clusters to get some of the major tributaries but that are still wadable streams, so we can use the same methods in these streams and streams that are close to project sites. So the Academy and Stroud sampled these integrative sites in 2013, and we'll go back again in 2015. The project sites, once projects were funded, which was funding from William Penn Foundation, went to the National Fish and Wildlife Foundation as well as the Open Space Institute. They released requests for proposals, and proposals went through review from their advisory committees and were funded in 2014, and there will be another round in 2016 and 2017. So we've so far done the project sites funded in 2014, and I'm not going to use the hyperlink, but this is a map of all of those project sites, and if you can see, they're labeled by the type of sampling and who has been sampling there. So this shows not only the number of sites, but the diversity of indicators being collected by different groups. And I'll go into this a little more later.
And if I could just interrupt for a moment there, folks, that mapper that Steph was just showing there is available on our website, on the urbanwaters.gov/Delaware website, under the Water Quality and Quantity Community of Practice. So if you're curious, you can find that there and call it up on your screen as well.
Thanks.
Okay.
Yeah. Yeah, and you can, so this is just the legend. If I go over here to content, you can add other layers to it. So as an illustration, I've got the AmNet New Jersey Ambient Biomonitoring Network stations loaded that show the quality of streams. You can add other ones from your computer or from the web, so with these base layers and other things. And we've also, got a point source layer, which may take a second to load. So as we accumulate GIS layers that we find useful, we're putting them up here so other people can use them as well. Okay. So the study design we're using at the projects is a BACI design, which means we're sampling before and after the project is implemented at control sites, which would be not influenced by the project, and impact sites, which should be downstream or impacted by the project. This is very intensive for time and effort, but we really think it's the best design to capture the changes at each site so we can tell the story at each project, and also to bring together as a comprehensive data set. So in 2014, that was the before control impact for projects funded last year and this year. And then we recommend ongoing monitoring after project implementation, which will be the after control impact. We also have some control sites that are not just upstream of the project in case our upstream site gets affected by something else upstream. We have some additional control sites that are also being sampled, and some sites for special research topics, which I'll also talk about. Okay, so now I just want to tell you about the tiers and the different methods that classify the tiers. For Tier 1, this is sampling that is mainly being done by the Academy of Natural Sciences and Stroud Water Research Center, but some of our partners can do Tier 1 for macroinvertebrates and chemistry as well. So for macroinvertebrates, we're sampling with Cerber samplers and sampling in riffles, which will allow us over time to be able to measure changes in this habitat. And you may know that riffles tend to be the most diverse habitat, and that way you get sort of a stable type of community to look at over time. We think quantitative sampling is really important for macroinvertebrates as well as algae and fish, because we don't want to just show who is living in the river right now, but we want to show in what amount and in what density to see those small changes over time. For algae, we're collecting diatoms and soft algae, and for fish, we're using multiple pass depletion sampling, where we aim to collect every single fish within a reach. The algae protocol is called SWAMP. It's a multi-habitat sampling protocol developed in California, if you're interested in that protocol. An important part about the macroinvertebrates that's very different from other groups is that we really encourage spring macroinvertebrate sampling. A lot of our really sensitive taxa, which include mayflies and stoneflies, are emerging very early, and so those summer and fall sampling events either don't capture those taxa or they capture them when they're so small they may not be able to be identified to species. So this is really important too for getting down to that species level and genus level for identification. This graph just shows for several different mayflies when the hatch is. So they become adults at these areas in dark gray. So you want to catch them before they become adults. And to get them all, it's best to go in spring. For Tier 1 still, when we think about water chemistry, these are reaching levels that we would reach when analyzing water samples that are sent to us by people who need to report back to the New Jersey DEP. So very low detection limits and high quality analyses. For habitat, we're using a modified US EPA Wadable Streams Assessment. So that includes that rapid biological protocol habitat assessment that a lot of people know, as well as a riparian forest index. So that's Tier 1. For Tier 2, one of the biggest things that distinguishes it from Tier 3 is that there's training. So the people who are doing macroinvertebrates for Tiers 1 and 2 are going to go talk to John Jackson, go out in the field with his group. If they're going to do fish, they're going to come out with the academy group. And the training part is really important so that we can assure the quality of the data. So for Tier 1, we use a Cerber sampler, but for Tier 2, they may use a kicknet. And we really found the New Jersey DEP's volunteer monitoring program helpful because they've developed manuals for high-quality certified volunteer monitoring. For water chemistry, they may use a lab with higher detection limits that aren't as strict, or they may use field kits. And then if a group were catching fish, a Tier 2 definition would be a single pass or trout presence absence. So these would be useful engagement groups. So it would be useful in outreach, but also this yields high quality data when we make sure that there's training in QA/QC. And for Tier 3, we don't expect, this is from watershed associations that are really more about outreach, getting people aware of what their rivers are like and getting people out there, but we're not with as strict of quality standards. But the thing about Tier 3 is that the people who are involved in this are often numerous and enthusiastic, and we don't want to tell them that their data aren't important. So we want to make sure we use this information as much as we can, but then if a researcher comes along and says, you know, what's going on with this data, that we can say, well, this is important for outreach, but not maybe at the level for your analysis. So just going through the summary of this, there's some overlap, and again, the difference is in training and quality assurance, quality control. So going back to this map here, the yellow are Tier 1 Academy and Stroud sampling, and the orange are cluster groups, and those will be a mixture of Tier 1 and Tier 2. So we can zoom in and click on them and see what the name of the site is and the name of the river and what cluster it's in and who's doing the sampling there. Another way we can use this tool, and we hope our partners will, if you see me, I'm just zooming in, hopefully not making anyone dizzy. Oh, I lost one. Here we go. You can see the streams here, so we have run some analyses. It basically is a screen over the stream data. Sorry, I'm just going to, I did this earlier, but it didn't stick apparently. So you can see the streams. These are the areas that drain directly into them. So if I can click on that, it tells me the watershed area, the stream order, and the different land uses in there. So this is something else we think will be helpful to our partners and our analyses. Okay, back to PowerPoint. So we're working closely for QA and data management by providing training with our partners, Stroud, and for example, Wissahickon Valley Watershed Association is another group that provides training for people taking out volunteers in habitat assessment, sampling. Another way for QA/QC with these groups is comparing lab analyses of water samples and making sure that the data we receive are within reasonable ranges. And we're also in progress of seeing how volunteers can collect Tier 1 macroinvertebrate data, but then hand that over to pros to identify. And I guess the last point I want to make is just that we're really happy to have so many partners that want to get out there and help us sample and get a lot of data. And so our objective is just to make the most of these data, to get people the training if they can move from one tier up to the next one, and to get this information into our database. So I'm sure many of you do this too. We've got our field sheets with these pretty good drawings of the stream, and then we put it into our Access or our Excel files, and I'm guilty of this. I just want to work in Access and Excel. But this is hard to turn into analyses quickly, and it's hard to communicate, what does any of this mean to people. So we've been working, and we have a database manager here, Scott Haig, who is working to develop a database that's open source and therefore can plug into other databases to make the data useful to us and to other groups. So we have all these people collecting data. We are QAing our data, but we assume that other people are doing their own quality assurance. So then we make sure there's geographic information connected to them and put, our knowledge and our research together. Make sure that people who are participating on the ground or monitoring are getting this information in a way that they can use to reach out to landowners, to talk to their county conservation district, or however they might find it useful. And then we are working with our collaborators to generate research, and then going back in and getting more data. So it's an iterative process, and we want to make sure that anyone who's interested in getting this data can get it, and we also appreciate how agencies and the Water Department and other academic groups have shared their data with us. I think I'm behind schedule, so I'm just going to quickly say that we sampled a lot of sites for a lot of things last year. I'm just going to skip over our research projects for now, but be happy to talk more with folks. And just try to show you quickly some of our results for macroinvertebrates. So these are color-coded according to the sub-watershed cluster. The green are preservation, and these other clusters are restoration clusters, labeled here. The data we collected we used to calculate a macroinvertebrate IBI called the MAIS. So what we're not seeing, and we're not too surprised, but it's good to note that all of where land preservation is happening are not all in the good range. We have a variety there, and the same goes for in the restoration areas. They're not all fair or poor. There are some streams that are good quality. So that shows that we're really capturing what are the typical conditions throughout these areas, and we have an idea of what to compare to. And then in Philadelphia, they tend to be down in the poor category for macroinvertebrates. For algae, as I said, they can be used to relate to specific nutrients and contaminants. So here, the y-axis is the percentage of nitrogen-tolerant algae, and we're seeing groupings of the preservation cluster sites with a low proportion of them, and the restoration sites with higher proportions. And this is the same with phosphorus where we see some grouping, but it's not quite so clear-cut. So as we're going through this 2013 data, of course, we have even more questions, and we're excited to see how the 2014 data help us answer more of these. For the fish IBI, so this is using the New Jersey DEP IBI for our data, and so we see that there are some excellent streams across and good, and here it doesn't paint such a dire picture for Philadelphia, which again points to our need to sample more than one indicator. Here, this cluster analysis shows how the assemblages characterize sites within each of our sub-watershed clusters. So the Schuylkill Highlands and the Middle Schuylkill have similar assemblages, and they're closely related to the Brandywine Christina. And then Upper Lehigh Poconos and New Jersey Highlands are similar, and Philly stands apart. So we're working towards interpreting these results a little more, and also in communicating what we're up to and how people can get involved. So we do this through reports. We have a newsletter, and I have some of our contact information in the last slide. So if you're interested in hearing, we have a monthly newsletter that just gives monitoring results and other things we've been doing. The web mapper, which you just saw and which Michael said's up on the website. Our database is in construction, and we hope that it can start to be used by people on the outside by the end of this year is what our goal is. And then Carol is working with government relations and reaching out to folks within your group, for example, and other agencies. And then every month, the third Monday of the month, we have a Tapping Our Watershed seminar at National Mechanics in Philly. So here's Matt Earhart giving a talk there. And I'd like to tell you who our next person is, but I don't have it right handy. But we will. So again, our objectives are to show changes from these projects, to do the monitoring, to use the monitoring data so that it can be used in outreach and decision making, and to work collaboratively with all our partners. And we'd really like to thank the William Penn Foundation for this investment, which is really a commitment to making sure that restoration and conservation can be done better in the future by collecting data on the effects of them. Our collaborators at Stroud Water Research Center and here at the Academy in the chemistry section, and all of our cluster partners have been essential to this project. And so here is our team. If you want to contact myself or Carol or Roland, and they can direct you to the right person. And here are some of the links to the web mapper and to our website, and of course our Twitter account. And I just found with Carol's help, our next speaker is on Monday, March 16th. It's Adam Levine, who's a historical consultant at the Philadelphia Water Department, and he'll talk about the history of Philadelphia's lost streams, which we know can be called sewers now. Thanks. Yeah, it's at 6 o'clock at National Mechanics on 3rd and around Chestnut. And it is a bar. Yeah, we called it Tapping Our Watershed for a reason. So that concludes what I have to say, and hopefully our goal is for this to stimulate conversation with everybody. And so I think now I will hand the mic over to Carol.